Thanks, Nanopore, for inviting me for today and also giving me the opportunity not only to be here on the Biopharma Day, but also in the last 2 days on the NCM meeting, which was already thrilling and amazing to see the new developments of Nanopore and also the applications by the community. And I several times had the feeling like measuring with Nanopore in the Antarctic or in wastewater or, I don't know, by untrained operators makes it probably a bit boring to just measure one or a handful of mRNAs in a fully equipped lab, like with trained personnel. So I'm really happy that you are still motivated to support us in this challenge. As Veronica said, I'm from a young and small biopharma company called BioNTech, located in a small city in Germany called Mainz. And the vision of the company is to use the power of the immune system to fight cancer and different diseases, mainly infectious diseases. And we believe in the power of science and in the power of innovation. So I think this is what makes us a good partner for Oxford Nanopore as well. I want to show you a bit of where we come from. So it's also, when Gordon showed the timeline of Nanopore, it looked quite similar to our timeline. So BioNTech was founded in 2008 by two more immunologists, Ugo Shahin, his wife Özlem Türeci, and Christoph Huber, as a spin-off from the Medical University of Mainz. And already before, they had some really important discoveries on the usage of mRNA to express certain antigens in cancer cells. And basically the major goal of founding this company was to develop a individualized cancer vaccine for the treatment of cancer patients. And actually only 4 years later, as you see in 2012, there was the first clinical trial started with individualized cancer vaccines. First, just naked RNA, a bit later also formulated RNA. And in 2017, there were the first readout and showed that this kind of strategy, that this treatment with individualized RNA is really making a difference in the treatment of cancer patients. Then in 2019, there were two important events in the company. First one was that BioNTech went to a public offering of stocks at the NASDAQ. And probably not that much important, I started at BioNTech in 2019. And just in time, because 1 year later, as you all know, there was the COVID-19 pandemics, which changed probably the lives of many people, but also the life of, the life of BioNTech. So actually, until that point, we were really only focused in cancer vaccines, so we didn't think of infectious diseases. But Ugo very quickly realized that the mRNA platform is the way to deliver quickly also a vaccine against infectious disease. And only a few days after the genome of the SARS-CoV-2 virus was published, we came up with some candidates, like some drug candidates, in January 2020, and only 3 months later, in April, we started a clinical phase 1 trial. And again, only 7 months later, in November, we could, in the first interims analysis, report that more than 40,000 subjects were vaccinated with our candidate, and we could show more than 90% efficacy, which then led to the FDA granting us only a few days later the emergency use authority, and a bit later also MHRA and EMA was giving us conditional approval for our cancer vaccine. And this was the one part, like kind of the discovery and driving it through the clinics. But in parallel, or maybe traditionally, you would wait until that point, and then you would probably ramp up the production, and then you can, slowly go into the market. So we did that at the same time, like we ramped up the production capabilities from actually milligram to gram scale at the beginning to a kilogram scale of RNA at the end. And by now, we delivered more than 4 billion doses of the community COVID-19 vaccine. So as I said, that was basically life-changing for the company. Of course, we earned a lot of money, and this is what we now invest into again in our cancer programs. But also, we learned that also infectious diseases are a good target for our mRNA therapeutics. So you will see that we have programs in both fields. So this is a bit BioNTech in numbers. So we have so far only one commercial product. We have more than 35 candidates in oncology and in infectious diseases. We are still a rather small company, but this number there, you have to keep in mind, when I started at BioNTech in 2019, I was more or less the 1,000th employee. And then within the last 4 years, we ramped up to more than 6,000 employees, which is challenging, I have to say. But which is also nice to see is that nearly half of the staff is actually working in R&D. We are spread all over the world. We can nearly say our hub is in Mainz, as I said, in the middle of Germany. And we have basically 4 GMP production facilities, 3 in Germany for RNA production, ranging from small scale to middle to commercial scale, as I said, kilograms of RNA. We have here in the US in Gaithersburg a GMP-compliant cell therapy facility, and this is already ongoing, but still a bit in the future. We are trying to put manufacturing labs into containers. So this is the BioNTainer, as you see down there. So it's basically RNA production formulation in standard containers, which we can ship to the place where we want to have it. And we are testing that in Rwanda, Kigali, and in Melbourne, Australia. I have to say that we are not only an RNA company. As I said, we want to use the immune system, and RNA is one way to do that. But we also use cell therapies, protein-based therapeutics, and small molecules to do that. But back to RNA, there are 2 strategies we are using to tackle the cancer. One is our individualized program, which is called iNeST, which means individualized neoantigen specific therapy, which means that we take blood and a tumor specimen from a cancer patient, we sequence them, and we identify so-called neoantigens, so that are antigens that are in the tumor but not in the healthy cells. And we put those antigens on an RNA, formulate this RNA, and deliver it to the patient. So basically, the patient gets a medicine that is targeting only the cancer, but not the healthy cells. You can imagine that producing for one patient needs very small batches, so we are talking about 100 milligrams of RNA. But what is crucial is to be fast, so we want to have a really short turnaround time. And I think I'm not at the latest number here, but we are talking about like a few months to deliver that. On the other hand, we have the FixVac strategy, where we use common cancer-specific antigens. basically an off-the-shelf product with much bigger batches. And there, the challenge is sometimes that there are different combinations of different RNAs in one drug product. As I said, oncology is one direction. We are also enhanced a bit the infectious disease programs. So, as I said, we have our COVID-19 vaccine, which we are annually kind of adapting to the prevalent variant. We also aim to improve the whole antigen, the whole vaccine, either as a standalone or in combination with influenza vaccine. Then we have herpes simplex virus program and shingles program, and very prominently, like one of the very deadly infectious diseases, malaria and tuberculosis. We are in phase 1 clinical trials for these two diseases. And I want to mention one, this is in the list there, which is mpox. We started a phase 1 clinical trial 2 years ago in 2022 when there was an outbreak in Congo. And there was even a threat that this could turn into a pandemic. So we were again launching a little light-speed program to be very quickly in the clinics. Then infection numbers were going down. But actually this year we see a relapse there, and we have, I think, in this year more than 15,000 infections and more than 500 deaths already. So we are pushing this program to be in the phase 2 or phase 3 clinical trials in the next year. I was talking about the application of mRNA, but let's have a look at the drug substance mRNA. Basically, we are using this elegant mechanism of the nature. DNA is transcribed into RNA, is then translated into protein. The protein has to function. The only difference is we don't do the transcription in the nucleus, but we do it in a test tube, in an in vitro transcription reaction. Test tube is probably not the right word. Probably we start with the test tube in the preclinical phase. But then, as I said, we are ramping up, doing it in a fermenter in a liter scale to produce kilograms of RNA. The RNA has some crucial features. As you probably all know, there's the cap and the poly-A to protect the RNA and to enable the translation. We have UTRs to regulate the translational efficiency, and the open reading frame that is coding for the antigen. And then, since the authorities realized that mRNA is a potential drug substance for the future, the USP is working on a guideline how to release test mRNA therapeutics. And I just copied here the list of parameters that everybody should test. And also USP is giving some suggestions how to measure them. Very quickly, identity, of course, to make sure that the molecule you have in your tube is the right one. RNA concentration, we don't have to talk about that. mRNA integrity or intactness means the percentage of full-length RNA molecules, which is sounds easy, but it's not so easy because of course it depends on the sensitivity of your method. Like the more sensitive it is, the lower your integrity number. So sometimes you have to argue a bit with the authorities that the lower number doesn't mean that your product is worse, but your assay is probably better. And then there's a whole list of impurities we need to measure, and some rather easy physical things like pH and appearance and so on. So you see from this list that it's very long, that there are different methods, different devices, and of course all of this has to be in the GMP environment, means that it has to be validated, you have to train your people. So I talked all the time about BioNTech and not about Nanopore. So this is the point where Nanopore comes into play, because of course that is the challenge, or that is where we see Nanopore can help us. And we were using Nanopore actually already since 4 years in the R&D setting, like just to measure some RNAs to find out what it is. But now, like since last year, we are in closer communication to bring that also to the QC. And we were really thrilled to see this QC assay that is now going to come. And I just want to in my last 30 seconds, give some comments or some thoughts I have. Like, identity is a really good thing. We can measure any RNA without any sequence prior knowledge, so this is really nice. The thing is that, of course, it's really important to further improve the base calling accuracy, and which is also nice that we can also measure modification. Integrity, as I said, is really crucial and depends on the sensitivity of the method. The other thing, and this is also true for the other quantitative methods, it's really important for us to make sure that we have an accurate and precise method. And yeah, actually, I'm really looking forward to get the device, to get the kit, and test it soon, and to be in contact and communication with Oxford Nanopore to make it maybe even better in the future.